The Genomicon kit is delivered to you with two parts: a Genomicon box and some tubes and powder wrapped together with an elastic band. When your kit arrives, you'll want to separate the tubes and powder from the box. The bag contains LB agar. This is what you use to create the LB agar plates that you can then grow your E. coli on. All you need to do is just add distilled water to this mixture, so there's no need for other nutrients or minerals. You'll have to add the antibiotics that are included in the kit at some point later. One tube contains 2 epitubes with freeze-dried E. coli. You will grow these bacteria on agar plates for your transformation 1 day before you go to do your transformations. There's another tube with an antibiotic called chloramphenicol in it. You will add chloramphenicol to some of your agar plates so that only your engineered bacteria will grow. This second tube is full of freeze-dried DNA ligase. This is the molecular glue that is used when you are assembling your DNA. You'll have to add water to these just before you do your experiment. These 3 components should be put in your freezer as soon as you get this kit. Now for the Genomicon kit box itself. There are many different components in Science Hack kits, and keep in mind that there are many different kits available, and so the contents might be slightly different. The kit comes with magnets. Throughout the assembly, you use these magnets to separate the DNA from the liquid. This means that there is no need for a centrifuge. You simply place a magnet next to your beads and they will be pulled to the side. Watch the other tutorials to learn how to do a wash step and assemble your DNA. After you receive your portion of beads for the DNA assembly, and in between DNA assembly steps, you'll need to wash. This is the wash buffer. It's recommended that you transfer your wash buffer to about 6 microcentrifuge tubes that you can refill during your experiment so that you prevent contamination of the main stock. This will also help more participants to access the wash buffer. There are several tubes, and most of them have colored lids. These are the DNA parts you'll be able to use in your assembly. In order to understand them better, check out the sequences in your Synbiota project online. You'll find a complete list there of all the parts available to use in your DNA assembly. This kit comes with pure water for dissolving the DNA and ligase. The DNA comes freeze-dried, so you need to add pure water to each tube. Check your Synbiota project for a full list of DNA parts and the volume of water that you need to add. The tube with the letter E on top is the elution buffer. Once you've completed your assembly, you use elution buffer to separate your DNA from the beads. If you want to store your DNA for a long period of time, or want to run some of your DNA in an agarose gel, add some of the buffer with an N on it to neutralize the pH of the DNA. After your DNA is isolated, you need to put them into E. coli bacteria. To do this transformation, use the tubes with the letter T on them as well as the LB growth media. There we have it, folks, the Genomicon kit. Get ready to hone your bioengineering skills.